So now we get down to mitosis, and mitosis has four main phases in it you'll learn about: prophase, metaphase, anaphase, and telophase. Collectively, we refer to them as PMAT, and the goal is at the end of mitosis to have two genetically identical nuclei. Once again, you can see a cell spends most of its life in interphase and very little bit here in mitosis, and then cytokinesis at the end. And hopefully you'll be able to see how these four phases all play in a key role of making sure you have that exact genetic copy when you're done. Prophase is the first phase in mitosis, and one of the real key things as we look at copying genetic information is this idea that chromosomes supercoil and condense. Normally the DNA is just hanging out in the cell because when it's nice and loose, it's much easier to replicate because you've got the single strands there. As it gets ready for prophase and cell division, it finds little proteins called histones to wrap itself around and becomes really condensed so that you can actually see the chromosomes. And if you remember from the onion root tip, you can actually see some of those chromosomes lining up there. And so in this picture, we've got little bead markers to represent our chromosomes that are going to split up. And this cell has 4 sets of chromosomes. And the next thing is, in order to help make sure that each side gets an even split, an exact copy of the genetic information, these spindle fibers start to form around centrioles, and that's going to be what helps pull things apart. The last thing is, we know in eukaryotes that there's a nuclear envelope that contains all of the genetic information, and that's going to have to break down slowly if the chromosomes are going to be able to split to each side. So in prophase, one of the big things is getting these chromosomes to form from just that chromatin or DNA, the loose DNA here. So sometimes this is called chromatin, and forming into more condensed chromosomes so that it's going to be easier to divide. Chromosomes. One thing you'll want to make sure you keep in mind before we move on is right here, this is your chromosome. It has genetic information. And remember, in the S phase of interphase, things were duplicated. So the one on top here is an exact copy of the one underneath it. So we call those sister chromatids because they're exact copies. And so trying to go through this progression, you've got chromatin, and it forms chromosomes that are made up of 2 identical sister chromatids. In metaphase, you've got your chromosomes here made up of 2 sister chromatids, 1 and 2. These line up in the center of the cell, which is sometimes called the metaphase plate. And those spindle fibers we talked about are now on each side here, and they attach to the centromeres of each of the chromosomes. And then this way, we can make sure that the top one not only gets 4 chromosomes like it needs, but it gets the blue one, the red one, the yellow one, and the green one, and that the bottom one gets an exact copy of the blue, red, yellow, and green. Now, chromosomes aren't really that color. We just did that to illustrate a point here. But the idea is you can't just randomly take 4 chromosomes on each side, or else you won't have all the genetic information the cell needs. It needs to be nice and orderly so that there's an exact copy and similarities in each cell. In anaphase, the sister chromatids here start to get pulled apart. So you can see that this genetic information is shared on each of those. They're identical. And each new cell, the one on the top and the bottom, will get an exact copy of that. And so in anaphase, they start to pull apart. And it's those spindle fibers we talked about pulling those sister chromatids from the chromosomes apart. In telophase, or some people pronounce it telophase, the nuclear envelope is going to start to reform here around the new genetic information that's on each side. The chromosomes uncoil and start to become no longer visible. In this picture, they're still visible. And the spindle fibers that pulled them apart start to break down. And going back to what we had talked about earlier, notice there are 4 chromosomes here that are each made up of one of the sister chromatids now, with the other identical sister chromatid being in what will be the other new cell. And then as you get back into S phase, those sister chromatids duplicate again, but those are one chromosomes made up of one sister chromatid. So to summarize the whole big picture, we'll start here with G1, although since it's a cycle, theoretically you can start anywhere. Notice you've got the chromatin in here, which is the genetic information. It's not condensed into big chromosomes, but there's only one of each of the copies of genetic information there. In S phase, they're all duplicating here so that you have 2 copies, because you're going to need 2 copies when you go to split. We have interphase, G2. Then we start to get to prophase, where now you can see these chromosomes are nice and big and thick, unlike over in the G phase. In metaphase, they line up on that metaphase plate, getting ready to split. On anaphase, they're starting to split. Along the middle there, you can see the separation. In telophase, the nuclear envelope is reforming, and you have them starting to go back into the chromatin, where they're not this thick, stubby chromosomes like over here. And then a couple different things can happen. It can just go back into interphase after cytokinesis, or you could go into G0 phase, which would mean there's no more divisions. And some cells tend not to divide anymore down the road after they've been specialized, like nerve cells and things like that. So that is a summary of the process of mitosis that you learned about. Remember those 4 key phases: prophase, metaphase, anaphase, and telophase. Spell out PMAT.